You can almost see their little tentacles on them. So, but
Great, thank you.
yeah, thank you. That's a good.
Can you define commensal for me, please?
It's an associated animal that benefits from its relationship with one animal, and the other animal benefits in return. So
is that different than symbiotic?
it's kind of the same thing. They both, they both kind of benefit. We use.
If they both benefit, isn't it mutualism?
Now you guys. Okay.
Oh, sorry.
I'm gonna stick.
Sorry.
I'm gonna stick with an associate because we actually changed that in the Okeanos thing because we were getting so confused about the terminology. We call it associate.
There's my parking stall here.
Quit using
Associate coral.
let's see if I can.
Cool terrain.
All right, here we go. You ready?
Here we go.
Symbiosis is a close relationship between two species in which at least one species benefits. Mutualism is a symbiotic relationship with both species benefit. Commensalism is a symbiotic relationship in which one species benefits while the other species is not affected.
That's right.
But we often don't know.
Yeah.
Something like that, so it's better to just leave a generic term like associate. It is associated with that
There's
animal.
some kind of symbiosis going on. Would
Yes,
be the generic, genericist, most generic of them all.
Right.
So a commensal would be those things that crawl up onto corals, like the stars or whatever, not to eat, but to feed in the water.
Yep.
Cool.
I would say so.
Thanks, Chris. Yeah, I found that one in the ID.
Did you find the black coral, the associated black coral?
No. Sorry, the one you just showed me.
Okay, I'm going to give you this black coral ID here. Okay, it's Antipathy silospongia.
Got it. Thank you.
Yep.
You said that was a black coral?
Yeah, that's a black coral.
Oh, nice.
So why are there all these different shelves, like lines and everything? Are they each an independent flow from back when this was erupting, or?
This isn't basalt. The belief is that this is carbonate. This is reef growth, and you're going to get pulses of reef growth associated with glacial phases and interglacial phases, where the seawater is actually rising or it's sinking, depending on whether more of the water is being bound up in ice or whether the ice is melting. And so you can get these terraces being formed.
That sounds more like erosion than other things.
Well, erosion is definitely involved in it, but I think that's why you often sort of get these sort of steps on a lot of these seamounts. And that's what I have always heard.
Wild.
Wow, look at the Argus view with those striations of the rock.
Yeah.
It's so wild.
I think they're showing the terraces right there. You can almost count them.
Yeah.
Probably each of those is separated, say, by about 10 to 15,000 years.
Wow.
Would be my guess. That's wild guess, of course. And these are Cretaceous terraces. And who knows what's happened back then, or at least.
But sometimes they blend together too. Like look in here, just off to the right. That's where two terraces meet.
Yeah.
Or not quite meet, but you know what I mean.
Right.
Get closer.
We're gonna rename those little sand piles as Trevor's Sediment Terrace.
Another one of those little potential Canthagorgia that we just sampled a little while ago. Bunch of dead sponge stalks down there in the sediment. Pretty fascinating geology here. Very different than what we were seeing down on the ridge itself.
Really different. Yeah, you could definitely get a whole sample right there for sure. That's what you're thinking.
I'm pleasantly surprised by this. I didn't think it would be so dramatic when we were creeping up on it.
Yeah,
Totally.
I know.
It's nice.
So what are the contour intervals in the HYPACK display?
10 meters.
10, really?
Yeah,
Wow.
unless they pulled a trick on me, I can check. Bridge nav, 20 meters, 140.
Shark behind Herc.
Shark behind Herc. Sneaking up on you. Yeah, a little sponge hanging out. Tiny little.
So it kind of answers one question. John Smith was wondering whether this was a volcanic structure, but I think the consensus from the science chat and others is that's not, this is a carbonate structure. So it must have been some type of little reef thing that was growing on the top at one point millions of years ago.
I just don't know why it's black. I mean, what's this from aging or just from the crusting?
Yeah, crusting. Well,
there's the scratches again.
I have a piece of manganese crusted basalt at home that I take sometimes for little public displays, and I also have a piece of manganese crusted carbonate.
They look the same?
Yeah.
Oh, wow.
You just break them and say, let's take a look at this,
zoom in on
please.
the coral, please?
Yeah. Uh-oh.
Uh-oh.
See.
We already sampled him.
No, that's the Solandaria. That's the hydrozoan, so that's good.
Yeah, never mind.
Those are Europtychus, or the crabs, I think, in the family Chirostylidae.
Bridge nav, another 20 meters, 140.
Oh, could we look at the shrimp? This is a different shrimp
you
up
bet
on
we
top.
can. Sure can do.
Yeah, this is new. We haven't seen him yet. I don't like the... Andy, quit messing with my teleprompter.
There's another kind of blue circle.
I like yellow, see? That's better.
That one's not drunk at all.
That one's striated shrimp.
Chill shrimp.
I know what you are. Oh, okay, I got to remember. This is Heterocarpus, and it's Heterocarpus... can't remember. There's a Heterocarpus ensifer and Heterocarpus something or other, and it's the other one. So that's the first one of those. That's very interesting. They sit on the bottom. They don't swim, just to let you know.
This is a wall. This is still a wall.
Oh, Laevigatus. Heterocarpus laevigatus.
Here's a star for you.
Here you go.
Just kidding, that's sand.
Thanks, Chris.
Star-shaped sand. Mess with my head.
There is exploration of, some people tried to see off of the island of Oahu whether there are enough of those Heterocarpus shrimp to be a fishery, a deep water shrimp fishery.
Yeah, sure. Yep.
What is the
Look
little
at these
white?
layers. These layers are
crab?
just...
Or is that just
What was that?
white?
White?
Is that just
Oh, I don't
broken coral?
Don't know.
Do a snap zoom there, please.
Yeah, just
What are you?
coral fragments, older
Yeah.
coral fragments. A little
Cool.
sponge.
Okay, come wide, please.
Yeah,
Nice
that's
dead
it.
leaf sponge.
Yep. But these ledges are just beautiful. Look at these
Really
things.
amazing.
We're going to see a dinosaur bone any second here.
Fish.
Yep, little fish.
Where?
Running along the ledge. Oh, he's going off screen.
Gone.
Gone. Oh my God, it's eerie.
So the thicker
Oh,
the
look at the
ledge,
Argus view. Whoa.
the thicker the ledge, I think, the longer the time period between some type of change in sea level.
Hmm.
Yeah, so it would be, so the coral had a longer chance to keep up with the island sinking.
Oh.
If I'm understanding things properly.
That looked like
Okay,
a heart in the rock. Jason is asking a question.
Okay.
Would a manganese encrusted sponge hold its shape?
Yeah, it would. So sometimes we can actually identify what the sponge is, even though it's manganese crusted. But if it gets really thick on there, then it would kind of start to look like rock. But it seems like most of them we've ever seen are kind of dusted. They have this very thin layer, unlike the rocks around them. But also the rocks around them are a heck of a lot older. So.
Ooh, what's under here?
Oh, what was that? That's.
Oh, you want to go back up?
Yeah, absolutely.
Ah, fighting.
You can do it. You can. I know you can do it.
100% right now. Oh, we're getting there.
There it is.
There it is.
Oh, it's a spot. I thought it was some kind of weird coral. Let's take a look at it. Good.
Okay, zoom in there,
what's
please.
that to the left, though?
Ah, okay. Feria oca, so that's good enough. There's a little Walteria next to it. And then what was that orange thing to the left?
To the left?
Yeah.
Keep pulling out.
Right
Right there.
Oh, it's an anemone, I think.
Yeah, it's
Okay.
like an anemone.
Yeah.
Okay, zoom in, please.
No.
Bonk.
Well,
What?
it's a Anthomastus that's closed up.
Yeah.
Those are where all the polyps would come out, the little black holes.
Oh.
Cool.
Yeah, great. Thank you.
Okay, come wide, please.
Thank you very much for that. Appreciate it. Some type of whip coral, probably a bamboo. So what's causing that sort of black shadowing effect above the lasers?
I'm just looking way up. There's just no lights
Oh,
there.
There's a
I
cave.
see.
It's like an underhang.
I
Yeah, I'm
see.
definitely under rocks now. You can see an Argus view. Bonk.
Oh.
Yeah.
Oh, yeah.
Gonna go do some spelunking any second
Yeah.
here.
I'm into it.
Bridge nav, 20 meters, 145.
Bonk. Yeah, stop that.
Pricing Gorgidae. What's that? Gold. Ooh.
Dramatic shadows.
Ooh.
Yeah.
Another Canthogorgia, similar to what we sampled. What terrain? What amazing terrain.
It really is, yeah.
Yeah. I never would have expected this in this area.
There's a strange looking coral. I think it's a Paragorgia though.
Porcelanis? Is it the Paragorgia porcelanis? Porcelain?
No, you're thinking of the Pleurocorallium. So that's just white Paragorgia, white
yeah.
bubblegum.
A viewer is asking, wondering how many onshore scientists are watching at a given time.
That's a good. We actually don't know because a subset of them actually connect to the chat to make comments
Fish,
and
shark.
recommendations, and then there's a lot of them. That's a shark.
Different
Yeah.
shark, no white spot.
Oh,
Yeah,
yeah.
you're right.
Oh, that's cool.
Oh, what's up in this crack? Look at that. That is so cool.
Wow, it's all the way down, too, from the top. You can see where it's severed from the top.
Oh, yeah, that's cool.
You're just itching to go into a cave, aren't you?
Oh, man, you have no idea. Absolutely.
Wow, look at that shear. That's incredible.
There's something in there.
Little coral.
Shrimp.
Oh, there's something bigger in there.
Yeah,
Nice
that's
little
one of those-
habitat.
Focused flow, I guess.
Yeah, I guess so.
Cool. Look at that.
It's oriented up down though.
Look at how far back it goes. That is deep.
I'm going to
crack.
deviate a little bit. I want to follow this a sec.
It goes to the center of the Earth.
That's fine. I'll catch up.
Yeah. It's the ultimate rift zone.
Well, obviously, something caused that to split open, the reef to actually crack open. You never know. Again, since this is so old, could have been a massive earthquake or something that caused.
Right, yeah. It's weird shadows. It's very confusing.
But it's really interesting. Look at that.
It is.
It's cool seeing that, I don't know, the yellow coral near the edge there. That was cool. All right, let's catch up now.
Big angularis.
Oop, bonk.
Look at the Argus view is so cool, watching the ROV fly off the ledge.
Thanks for leaving it dark there, Aaron.
It's not much you can add to it.
The abyss, you mean?
Yeah.
Or this yellow coral.
Yeah, the
Bridge nav, 20 meters, 145.
And little fuzzy Feria oca too, with this little black coral associate.
Look at that. There's some columnar basalt, except it's the wrong way.
It's not basalt, it's carbonate.
Oh, that's what I meant. Yep, I just mispronounced it.
Columnar carbonate.
Basaltinate.
Basaltinate, yeah.
Pretty weird.
That is weird the way it just sloughs off in those columns.
That's cool.
What is that?
Fish.
Oh,
There's no way I'm keeping up with that boy.
Kevin Conrad says,
You can't if you don't try.
so we saw these large fractures in the carbonate walls in the corner rise seamounts earlier this year. His guess is that it's fracturing during subsidence.
Ah, okay. Thanks, Kevin. By the way, that fish was a halosaur. It was another
Can we get a reality check on heading?
Aldrovandria phylacra, but that's okay. Quick observation. So Kevin, is that a really slow process to create a fracture like that, or does it happen very suddenly and abruptly where the reef?
Still 145, all right.
Just,
You do? Okay.
just curious how that forms.
All right.
Bridge nav.
Said, great question. My guess is slow, but I'm not sure.
Step, 100.
I'm going to pop up on top for a minute, see what's up there, and then back down in a sec. To see as much as possible. It's just so much to see in life. It's hard to see it all.
Yeah.
It's very fun habitat.
Well, you know, there's going to be a number of guyots next spring when we do the Liliokalani seamounts that were mapped before this by the ship before this cruise in NA133, and this would argue that at least one or two of those dives ought to be done on the guyot tops. Guyot, looks like a white paragorgia.
Yeah. We got some more of those big corals up here.
Oh yeah.
They definitely like the top.
Yeah, they do.
Some good current flow up here. Is this another ledge here, perhaps?
Looks like it. Small terrace, I guess.
Yeah.
No, not so small. So I have another question for Kevin, if he's still on. So it doesn't look like the manganese crust on these terraces is botryoidal, and they're going to be younger, of course, than deeper on the seamount, I would guess. But would they be that much younger, or can you just offer maybe it was exposed later and it hasn't had time to build up? It looks like just kind of a black coating. Whereas down below, we saw a very clear botryoidal surface or grape-like surface.
All right, finish that up. Do you want to wait for Argus to settle, or you want to start moving south again? Do another one out? South, yeah, okay.
Not significantly younger,
Bridge
but
nav, 20 meters, 145.
he said not significantly younger, but would be shallower for longer. Ideal depths for Fairmangan growth is around 2,500 MBSI.
Nice leafy sponge.
Yeah, thank you, Kevin.
Meters below sea level.
Oh, meters below sea level. I don't suppose, we have a couple minutes left, right?
We do.
You see one of these kind of cone-shaped sponges that are small on the bottom, and I'll circle one just like this one over here.
Yep.
Is there any chance you could break off a small piece and slurp it?
Sure.
Bridge nav, hold position.
Let's take one more look at it. They're curious. They're not quite like the ribbon sponges because they're actually forming little amphitheaters. I'm not absolutely certain they're the same thing. And I know for sure we've never sampled these kind of like Waikiki shell sponges. In fact, that's what we used to call them.
Waikiki shell sponge.
Shell sponges.
Right on the ledge. Right on the edge of the ledge.
The edge of the ledge.
Ledge edge.
Well, if you are a qualified PhD geology
Can you set up
candidate,
the cameras, please?
Kevin has been commenting in our science chat, Kevin Conrad, he's looking for a PhD student to work on the next cruise next year to go out on to look at the Liliuokalani seamounts cruise. So it'd be a 31-day seagoing expedition.
I was changing to camera, same
I think
section.
that's still, it's on your website, Kevin, so I'm not sure if it's still active,
please?
but looks like applications are through February 1st, 2022.
Yeah, let's get a good colony shot. And then
can anyone apply for that, or?
Okay, come
looking
out
to
a little
recruit a qualified PhD student. Yep, it's still available, Kevin Conrad. So look up, just Google Kevin Conrad, and you can find his info and see if that's of interest to you.
Okay, come wide, please. Yeah,
wide,
line
wide.
up bubble. Thanks.
Oh, okay.
I tried to snip, but in fact it just ripped. So we've got this much now. That ledge is causing some troubles. Zoom in, please.
Close quarters slurp.
Yeah, I'll say.
well, the other thing you could do is put it in a box with like the coral or something. That Canthagorgia, you're certainly not going to.
I think he's got it.
Oh, he's got it? Okay.
I have faith.
Okay, slurp 30, 40 something percent.
40%.
Pick a number. Yeah.
Come on.
Maybe more, maybe 70.
70.
Take another cut. There we go. Come on, you little rascal.
There you go.
Nice.
Might as well get the rest of it if I can.
Sure, yeah, please. And a moment of truth, will it come out in the jar?
I have to chop
There
it
it
up more.
is. It
There it goes in the jar.
It's trying.
Get in there. Get in there.
Maybe
Get in there. Come on.
it's maybe the last slurp of the night.
Slurp wars.
Come on. There you go.
Nice.
Yeah.
Yep.
Little mass.
Last little piece. Oh, that's
Oh,
a
that's
big
a
one.
big
Too big.
piece.
Oh,
What have you become? Get in there.
Oh, the big piece comes through.
wow.
Wow, good job.
Wow.
There it goes.
All right.
Impressive.
Come on, please.
Absolutely.
That was exceptionally fun.
Thank you very much for that.
Thank you.
That was-
Nice. Was that slurp jar 2?
2, yes.
Thank you.
And that was 1, 2, 4, yeah?
Yes.
Great, thanks.
I'm going full wide on her.
Thank you.
I'll finally answer the question whether we can call the Waikiki sponge the same thing that we call the ribbon sponge, or at least in
You
the
see them all
same
come in? I
genus.
think there's one more.
I think
Okay.
so, yeah. I only
There's
saw
one
Always
more.
wondered about.
I've seen 3 in there, but I think I stuffed 4.
Yeah.
I don't know. You can turn it off, let it sit for a minute, and then turn it back on in a
Okay.
minute. All right, can I get front porch too, please? Okay, science, do you want to do another step? One more, probably got time for one more ship move and
Sure.
arc move, and then we'll do the handover.
Yeah,
Bridge
that
nav.
sounds great.
20 meters, 145.
Maybe I have a dirty, rotten liar. We'll see. Okay, I try one more 70% or something. I think I see all 4 in there, actually. There's one up on the right there, too.
Yeah.
I don't know. I guess we'll never know.
Should I flush it?
Sure.
Okay. Forward.
So do you know what the fate of that, the pieces that, you know, I'll go home with a piece to look at it under the scope, but these demo sponges are hard for me. So I will take a whole lot of pictures of the spicules, and then they will get sent to the Netherlands, where a demo sponge expert, who's a really great guy, Dr. Rob Van Soest, lives, and he's helped me over the years with a lot of IDs on these things, because for me it's really hard to identify them.
Yeah.
But really good, good guy, extremely knowledgeable.
Hi.
Got some interesting habitat. All
Okay,
right.
to all of our viewers out there, thank you for
Hui Hua Kai.
viewing us
Leaving the
on
house.
nautilus.org. We're making a watch change right now, so please continue following us on nautiluslive.org.
Yeah,
Ahoy
thank
ho.
you. What am I doing wrong here? Let's see. Oh, there it is. Oh, yeah.
Check, check. Hear you, check. Negative. I am perched. DPL reset? Yeah, sure thing.
We're starting with the fish puns already.
Curious what the fish pun is. What is that?
Perched.
Didn't even get that.
Steve.
Wow. Pretty early in the wash for that, but okay.
Hey, I didn't say it.
I couldn't get to the groan button.
Whose turn is it next? It's a different bird. Okay, yeah, we're all set up here whenever we want to start with the...
Great, thanks. So as we do on our shift watches, we're going to go ahead and take a moment to ground ourselves in the work we're about to do. So I'm going to invite our control van to go ahead and mute your microphones. For those listening at home, we are going radio silent in order to conduct our private ceremonial protocol that acknowledges and honors the biocultural resources we're about to interact with and that are present and that we'll be impacting. So we'll be back with you shortly.
Nav, did you get some instructions from the outgoing team about what they were setting us up for?
Yeah, so from what I understand about this feature right here at the top, is you can see in Herc sonar, and then when they were below it, it's quite a rigid step. It's not very tall, but it's kind of a little pancake sitting on top.
Mm-hmm.
So they've been, I didn't get any instructions as far as like where to go beyond that, except we obviously have gone through, I think, the end of the planned end. So we're up here. It's quite a bit shallower. We're not 980 meters, so that's a swifter ascent. So we'll have some more bottom time. We'll do those calculations in a bit. But yeah, I don't have any extra places to go.
Okay. Let's see. I didn't have a chance to watch the video before coming up here. Do we know if they were having more luck seeing animals on the top of this pancake or on the side of this structure? Was anybody watching before we came up here?
The last thing I saw is they collected a sponge.
Yeah.
But it looked, no, I'm not sure. So you can see here, we haven't quite crisscrossed the top of it. It looks like they were going around the edge.
Yeah,
so
it looks
presumably
like they're following a contour.
Yeah, they might have seen some animals along there.
Yeah, Chris had said that this is a calcium carbonate cap. And so we could cross over if we were interested. They were having fun looking at fish species along the wall.
Okay. Jess, how do you feel about some wall flying?
Yeah, let's do it.
All right. So Rennie, if you could set us up to kind of follow this depth contour around this flat structure.
Last I heard, said it was 145. That was the strike, so
145.
we'll probably have to do some shorter moves. It's also shallower, so we don't have to wait as long, but in order to try to stay on that wall. So I'll do 30, 145 and see what starts to curve around or not.
Roger that. 30.
Yeah, you can see in the Argus sonar that Argus is looking over it, so that's kind of, the wall doesn't go up too much higher.
Yeah.
But you can see a little bit of it in the top of Argus's screen. They just pulled Argus off the wall.
Yeah, so maybe we'll, just as we're flying, kind of want to keep that view in Argus, see if there's animals up there that we might want to check out.
Sure thing.
While we're moving along this slope.
Yeah, I can just go up and down the wall.
Yeah, that sounds good.
Sure thing.
Should only be about an hour up.
Okay.
If we stay at this depth.
Yeah, so we're all getting our bearings here on this Tamana seamount. This is our watch's second time seeing the seamount, although last time we were facing a very different cliff as we swapped off shifts. Now we're looking at the top of this flat guyot seamount. And this is the shallowest we have been with the ROVs in this expedition, other than ascending and descending. Thanks for everyone watching at home. Lots of excitement about the things that have been seen. Thanks for the updates. Yeah, if you're interested in messaging us, you can use the message feature on nautiluslive.org. And for those hundreds of you following along on YouTube, you can, again, swap over to nautiluslive.org and drop your questions or comments in that chat feature, in the message feature, I should say. It's not a back and forth chat. I have a hot tip that there might be some large nodules up at the top here.
Thanks. Jess, I have a request.
Oh yeah, let's do it.
Would it be possible to slide out the forward bio box so I can see what it looks like in terms of space?
Yeah, sure thing. I'm gonna go ahead and pop that out there, Jake.
Yep.
Oh, slides.
Yeah, okay, great. There's a little space in there for me to pick up something else.
Roger that.
Thank you.
Sure thing. Thanks, Jake.
Yep.
We have some folks that have been with us since our watch hit bottom this morning. Thanks so much for being such an intrepid explorer with us today.
Impressive.
Yeah, it's exciting.
We don't even do that.
Yeah, we nap.
Play cribbage.
See a theme forming here. Eat, sleep,
There seems to be
play.
a little shadow at 12 o'clock on the Herc camera. Is that something on the vehicle, or is that just the way it's oriented?
12 o'clock?
Oh, do you mean the top of our screen here?
Top of Herc's main HD camera, there's like a persistent triangular shadow between the lasers.
Yeah, it's just a shadow of the lights there. Kind of. It's more dramatic if we go like that.
Yeah, right.
Okay, why is this not working again?
Are the porch lights on?
Porch lights are off.
Downlights are off.
Downlights are also off. Yeah, I think it might be blowing out the picture. You can try to put them on, but
No, it's okay. I was just curious because a lot of times we don't see that darkness, partly because we're tilted down a little bit, partly because we have other lights on.
yeah. Can you look down a little on the HD cam there, Jake? No, pop down. On the HD camera.
Oh, okay.
Okay, so I think if you just pause that forward movement there. I'm just kind of following what you've done here. It seems like in order to keep it Argus off, maybe a little less than like 135 or
Yeah. Or 120 or something.
Yeah, we'll keep Argus off a little.
Roger that.
That's right.
Yeah. Just so it seems like you'll still be able to descend, and we won't have Argus on top of the pancake, but might be out of frame without some camera magic.
Camera magic, yeah. Jake camera magic there.
Yeah.
Nope, it's not on.
So does this carbonate rock have a ferromanganese crust? Is that why the coloration is like that?
That is what it appears to be we are looking at.
Yeah.
Yeah, front row, sorry I'm not giving you much instruction. I'm trying to still get set up back here.
Oh, that's fine. We'll just looking. Keep moving along.
Steve, what was the first sample number of this dive?
That was 109.
Thank you.
Word on the street is they took a push core.
Yeah, they did.
Did it have any nodules in it, or was it just sediment?
No, it was in up on this carbonate reef, in between some big manganese encrusted blocks. There was like a little ravine with sediment in it. Collected a core.
Seems to be some stuff coming up in in Argus. Probably more of these coral.
Yeah, there's some corals in front of us, and it looks like maybe sponges behind us in Argus's view.
Oh, yeah.
Could we back up a little bit just to see?
Yeah, sure thing.
Thank you.
Seems to be, again, like the life is preferring the interface between the top and the wall.
These are all, I think these are all corals here.
Yeah, you're right. Good idea. Nope. Okay, whatever. That one's going to be where it wants to be. Okay, 114.
Steve, for the depth and the surface areas, is this region a bit less biodiverse than what would be expected compared to, say, off the U.S. coast?
West Coast, East Coast?
West Coast?
Typically, so the biodiversity here for the deep sea benthos is higher, for the megafauna we're interested in, so like corals and sponges. I would say, I would argue that it's higher here, although the mass of the biology is probably less. The west coast of the U.S. is in a very different oceanographic setting than out here. This part of the ocean is called oligotrophic, meaning it's very nutrient poor, the surface waters. And for that reason, productivity is quite low, which is why we can see and have great images of Hercules from Argus. There's not a lot of marine snow, not a lot of phytoplankton, and this allows us to see very far, but it's not very good source of consistent food for the deep sea benthos, which relies on stuff from the surface. But off the west coast, what you have is more of an upwelling regime, which physically takes deep, cold, nutrient-rich water, brings it to the surface, which fuels productivity. And that productivity translates into organic carbon that sinks down to the seafloor and drives very high biomasses of things like corals and sponges. But generally, it's not always the case. Upwelling zones are more have poorer diversity compared to these open oligotrophic waters, and there's also a tendency to, as you move further south towards the equator, have latitudinal gradients in diversity. So, higher diversity on the equator in the Western Pacific versus the Eastern Pacific, for example. So a lot of factors going on.
Yeah, thanks, Steve. Thanks for breaking that down.
Yeah. We're also at this water depth.
I think it just had like one tangent.
At a relatively low oxygen concentration in the water, which is another stressor for your animals.
Right, yep. And what's our oxygen right now? It's 40, oh no, that's not right. 42 micromolar, which is pretty low. Yeah, it's not hypoxic, or it's not it hasn't reached that threshold, which is about 20 to 25 micromolar, but it's quite low. In my own research, I did quite a bit of work off of Costa Rica as well, in seamounts out towards Cocos Island in the Pacific coast of Costa Rica. And out there, the oxygen minimum zones are quite intense, and you end up with very strong selective pressures across, as you increase depth across the oxygen minimum zone, that weeds out certain species of corals. And in fact, you have a full community turnover across a few hundred meters because of the oxygen mutations.
Hey Steve, Emil here in the lounge. Beyond the lasers, there's that patch of sediment there that looks like it has symmetrical ripples.
Yeah, I see it.
And if we could get a scoop sample of that sediment,
we need really good images.
And some good images of the ripples before we sample, that'd be great.
When you say scoop, what do you mean? Because a scoop is not going to recover very much.
And we have no
we just want to get the sediment sizes, the grain sizes.
Okay, we don't have anywhere to put it.
Yeah, there's no open boxes.
Oh, boxes are full?
Yes.
Yeah.
Could you slurp some sediment?
Is there any fine enough meshes? Yeah, I think the mesh we have on there now is 5 millimeters. So I have a feeling that everything's going to get sucked out.
Okay.
That's all right. If we just get some good images.
Yeah, images will be fine.
Great.
Okay.
Can do.
Lots of stills. And then if we can get this clip as a highlight.
Jess, if we could get the lasers onto the sediment with a closer zoom.
The video highlight of the. Yeah. We need, if they can move the lights to see, does that make sense?
Look at it.
Lounge, your mic is hot.
I just turned it off.
Thanks, Jeff.
Can we also get some Argus stills, please?
Got him.
Jeff, you want to push on in there a bit, please, for Herc? All right, go ahead and come full wide, please.
Want to go tighter than that, Lounge, or is that sufficient? Really see the rippling in the
Argus?
in Argus, yeah.
Yeah, the Argus shots look great. Nice job with the lighting there, yeah.
All right, go ahead and push on in on Herc there a bit, please.
Super. All right, thanks very much.
Yeah, sure thing, Emil. All right. Full wide, please. Oops.
Looks like we're okay for another 145.
Yeah, sure thing, ready. Thank you. Beth, do you know why the interest in the ripples?
I do not. I am not sure which member of the shipboard or shore-based team, what the intention is with those requests.
There's a big one up there. Two.
Did you want to look at the corals there? Or you noted?
These are ones we've seen before. These are genus in the genus Caloptrophora. But yeah, I mean, if there's time, you know, if you need something to look at, we can of course get some better imagery. But we can probably ID them from colony morphology. These are pretty characteristic of this depth, and while there are a number of species, you know, we have pretty good sampling of primnoids in this region. And in addition, yeah, we've got two really interesting comparisons here. These smaller colonies that we're moving through right now are Caloptrophora wyveli, which is the same genus as the larger fans we just passed. But interestingly, they form two different clades within that genus. The Wyvali clade, which has its polyps that retract downwards. So when the polyps close, they point downwards, whereas most of the other species in the genus kind of point upwards when they retract. So it's one of the characteristics we can use to really identify a lot of these sea fans in this particular genus. Did that loose sponge come from us?
I don't know.
Or okay. Just want to make sure it wasn't something that was sucked up. Didn't look like the last thing they sucked up. It did not look like that. Okay. Good. Great.
Right,
Thank you. Good captures. These primnoid corals, they're exclusive to the deep sea. They don't really have any shallow water relatives that you might find in a reef, for example. So they're actually quite diverse in the deep sea, and they're pretty unique to this environment. Well, we've been seeing some other corals today that I've been keeping my eye out for, like another species of coralliid precious coral, Pleurocorallium, is around here as well, and I think that's what the lasers are on right now. It's a white, usually pretty stout branching precious coral, similar to those red precious corals we've been seeing, but separate genus. We've also been seeing a number of yellow fans, which could belong to a couple different families, Plexaurids and Acanthogorgids, one of which we sampled, so should help narrow down some of the identification.